Hello everyone! Congratulations on making it to the end of the semester. This is the final lecture for cell biology. It's actually one of my favorites, so I'm always sad because we never get to spend more time on it. But today we're going to be talking about cell communities. So how do these cells individually, we've talked about how they function individually, but how do they function in groups? So we're talking about tissues here. We're also talking about stem cells. How do these communities or tissues arise? Right? We've got stem cells as the origins of tissues. And then talking about cancer. So one interesting way to view cancer, or one perspective, is that it is individuals in a community that aren't doing their part. They're not kind of getting along with others, not listening to the rules, things like that. So we've talked about cells, right, as the building blocks of the body. Cells make up tissues, tissues make up organs, all of our organs make up an individual. And we know that we have, just like we have a blueprint for a house or a building, we have a blueprint for the body, right? So our our genes are acting as a blueprint. And we also have, just like buildings, have various components such as plumbing, telephone lines, electricity that has to be throughout the house. Our tissues require things like blood vessels, nerves, immune support, right? So this is what we're going to be talking about. It's how tissue is organized, as well as how it's maintained. It's talking about stem cells. And then lastly, what happens when tissue renewal gets out of hand, which leads to cancer. So the first thing we're going to talk about, the first objective of this chapter is for you to understand what the components are of the extracellular matrix, what types of properties they can impart to tissues, be able to predict what types of extracellular matrix components you would find in one tissue versus another. Second, we're going to talk about how tissue is organized, and the example we're going to use is epithelial tissue. You're going to learn about cell-cell connections and how the different types of cell connections can promote different types of tissues and functions. Third objective of this chapter is that you know what the definition of a stem cell is, what it does normally for a tissue, and then also the different sources of stem cells for research and therapeutic use. You should be able to compare and contrast embryonic stem cells and induced pluripotent stem cells. Lastly, we're going to talk about when tissue maintenance kind of goes out of hand, when there's overproliferation and a cancer, what the effects of that can be on the body. So we're really going to focus on how it begins, and then we're going to talk a little bit about different treatments that are being tried out to cure cancer. So let's first talk about the extracellular matrix. And while it's not an organelle, right, it's outside of the cell, extracellular, we did have that as one of the topics in our very first cell biology assignment, where you guys had to look up different organelles and learn about them in more detail. So if you remember, that group talked a lot about collagen. We're going to talk about collagen today as well. So first of all, what is tissue, right? So there's some main categories of tissue in the mammalian body, right? So there's nervous tissue, muscle tissue, epithelial tissue, and connective tissue. And we have examples of those all here. You can see on the bottom left, we've got some nervous tissue. Here we have muscle tissue. This is an example of epithelial tissue, and here connective tissue. So tissue is composed of cells and any extracellular matrix, so any of the kind of surrounding materials. And so for short, extracellular matrix is called ECM. Now, where do these components come from? The extracellular matrix is secreted by cells that are in that region. Some functions of extracellular matrix. First, to give tissue strength. So you can think about like bones or tendons. The reason why they have their strength is due to components in the extracellular matrix. Versus other tissues that may be more squishy or elastic, they have a different type of extracellular matrix. It's also important for helping cells to interact or to bind to each other. So some cells will be very closely interacting and bind to each other directly and have very little extracellular matrix, whereas there's other tissues where the cells are rather far and few between. And so to hold that tissue together, that extracellular matrix is really important. When you're thinking about animal versus plant extracellular matrix, we have to consider the amount of movement that these organisms are participating in. So in animals, since animals can move, their ECM not only has to be strong, but it also has to be flexible. In comparison, think of plants that don't move very much or as fast, their ECM can be more rigid. Here we have an example of some connective tissue. We're looking at bone here, and the nuclei have been stained in black. And you see how the nuclei are rather sparse, right? These cells are rather far apart from each other, and a lot of this tissue in gray, a lot of the tissue is composed of extracellular matrix. You can also tell in this example that it's very organized. Connective tissue is going to include things like tendons, skin, bone, right? And so some of it's going to need to be very flexible, some of it needs to be more stiff, right? So depending on what the structural properties are required for that cell, for that function, depending on what structural properties are required for that tissue, the components of the extracellular matrix will vary. Now, opposite of connective tissue, and when we think about connective tissue, we have cells scattered throughout a lot of extracellular matrix. Epithelial tissue is different. The cells have very close connections, and there's very little ECM, right? So they're very close together. So we're going to talk about a few components that you should know of the extracellular matrix. The first is collagen, and this provides tensile strength, right? So what does tensile mean? If you think of rope, right? Rope, it's pretty sturdy, but it also has a little bit of flexibility in it, right? And so the more collagen you have, the more kind of tough, the less flexible the tissue is going to be. We also have elastin, as the word makes you think, elastin, elastic. This molecule helps to promote stretching of tissue. We also have sugar molecules that we'll talk about in more detail. These can allow some of the compressive properties of things such as cartilage. Fibronectin is a protein that is very important for helping cells interact with the extracellular matrix. So if a cell needs to crawl across a tissue, across extracellular matrix, it needs to be able to grab onto it properly. Now we're going to spend a good amount of time talking about collagen in this section, so I'd like you to really pay attention here. So there's a lot of different types or variants of collagen. I don't expect you to know those, but they do have some common properties. So as I said before, collagen is going to be found in bone, tendon, and skin. And the structure of collagen, the protein itself is a single polypeptide chain, but the polypeptide chain will then be kind of braided with two others to form a molecule of 3 strands. Then that triple-stranded molecule will be bundled together into what is called a collagen fibril. And then these fibrils can be further packaged together to form fibers. And so the fibers are what you are looking at in this electron micrograph. It's kind of neat. You can see some that are going across the section in kind of this longitudinal way. And then we also have some cross sections that are coming out towards you. And within these fibers and these cross sections, you can very easily see the fibrils. Right? So it's important to note that the fibers are made up of fibrils. The fibrils are made up of triple-stranded molecules. Right? Each of those strands is a single polypeptide chain. One thing that is easy to mistake is that collagen is not cytoskeleton. Right? It is not found inside the cell normally. Its function is outside the cell. It's made by cells, but its ultimate function is in the extracellular matrix. Here you can see some cells that are just kind of coated with this web of collagen. And cells, the particular cell types that will be secreting extracellular matrix components include fibroblasts. These are going to be found in skin, tendon, other connective tissues. And the cells that secrete extracellular matrix in bone are called osteoblasts. So these molecules are going to form kind of lots of clumps, right? Your aggregates or webs. And particularly in terms of collagen, let's look in more detail at what happens when collagen is secreted by a cell. Collagen is made in, as we said, fibroblasts. It can be made in osteoblasts as well. And when the collagen is first made, it is in a precursor form called procollagen, and it gets secreted by endocytosis. Now you can see that in this diagram, when the procollagen gets outside of the cell, an enzyme will cut off kind of these end regions of the collagen molecule. And now we have the mature form of collagen, and the collagen can then assemble into these fibrils. So we want the cell to assemble into these aggregates, these fibrils. Sorry, we want the collagen to assemble into these aggregates, these fibrils, but we only want that to happen outside of the cell. Right? Inside of the cell, clumping and formation of large masses of collagen would not be good. And so this molecule is only able to do that once those terminal regions have been removed. And so that is why this protein is first synthesized in a precursor form that will not be able to interact with other collagen molecules until it gets outside of the cell and exposed to proteinases. Here we have a little problem. In class, normally I would have you stop and talk with your partners. This is a drawing of a real person that had very elastic, stretchy, loose skin. So I'd like you to think about how this could happen based on what you know about the extracellular matrix so far. So we've talked about things like collagen, elastin, fibronectin, polysaccharides, right? In this case, this individual had a mutation that affected the protein structure, particularly of the protein collagen. We know that collagen is important for tensile strength, right? So kind of the tightness of the skin. We know that people try to get collagen reinserted or more collagen in their face to try and make it look younger, right? Because with age, tissue gets kind of looser, we get more bags, right? So in this mutation, the mutations in the collagen prevent it from forming fibrils correctly. And so because these fibrils aren't arranged in the proper orientation, they cannot provide that tensile strength to the skin that is required. And so it's going to be very loose and stretchy in this individual. So how should collagen be organized? So we've talked about these fibrils, right? But in addition to just assembling into fibrils, these fibers themselves are also organized in the extracellular matrix. In this diagram, this electron micrograph we can see of skin, the orientation is crisscross. So we have these several layers, and some layers you can see the fibers are facing in one direction towards us, and then in another layer they are perpendicular going left to right. Right? So this is kind of a crisscross arrangement of collagen, and that helps to really provide strength to the tissue. Right? The skin stretches in a variety of different directions, so it makes sense to have this crisscross shape. Now, other tissues don't really need a crisscross arrangement, such as tendons. Tendons are generally only going to be pulled in one direction, right? And so in that case, the collagen fibers are all going to be aligned in the same direction, in alignment with the force that is applied to that tendon. The way that this collagen is oriented, we know that the fibers will self-assemble, but the organization of those fibers, once they are formed, is performed by fibroblasts, so the cells that secrete the collagen. And so they will actually crawl along the collagen fibers and organize it. We can see here in this picture a heart explant, so this is kind of a clump of fibroblasts. These fibroblasts have started to grow out away from that initial chunk of tissue. They've started secreting collagen, and they've also started crawling along the fibers to orient them, align them with each other. So in this picture, each of these little spots, each of those little spots is a cell, right? I can't go back in this presentation. I don't know why, so we're just gonna keep going. So the next thing, an important component of extracellular matrix is integrins. All right, integrins, they are integral. They span the cell membrane and they help to connect the outside of the cell to the cytoskeleton inside of the cell. And we talked about fibronectin earlier, right? Fibronectin is also critical to this cell attachment to the extracellular matrix. So both integrins and fibrins are responsible for helping cells to attach to their surroundings, particularly to collagen in the extracellular matrix. You can see here we have integrin proteins functioning as a dimer, and they are spanning the bilayer of the cell membrane. And on one side, they're interacting with the cytoskeleton, the structural component of the cell. And then on the other side, they're interacting via fibronectin with the extracellular matrix protein collagen. Now, if these integrins were not spanning the plasma membrane, this connection would not be as strong. All right, you can imagine if the cell was not connected, say in this example, to actin, if the cell tried to move, there's potential that that integrin could just get ripped right out of the membrane. Kind of like if you're walking in the mud and you lose your shoe, right? If you don't have a nice tightly tied shoe when you're walking, you could potentially lose your shoe, and that's no good. Right? So this is why it's critical that this integrin protein span both sides of the membrane and that it also bind to cytoskeleton inside the cell. Your book talks about how integrins help cells crawl using a catch and release method. You can see that the conformation of this protein can change, right? It's dynamic. So when the integrin needs to grab onto the surroundings, it will kind of unfold and expose its binding site. And when the cell needs to let go, it will fold back up and kind of inactivate itself so that it can, the regions that bind to the extracellular matrix are not exposed anymore. Right, so just like when you're walking, you have to take a step forward, but then with your back foot, you need to lift up and let go. So the same thing is important for cells, to be able to grab on, but also to be able to let go. Just like with collagen, there are a lot of different integrins. They are going to, depending on the integrin type, bind to different molecules. So we just talked about integrins binding collagen in general, but we know that there are a variety of different collagen subtypes, right? And so integrins, there's going to be different integrins expressed on different cell types depending on what they need to bind to. Some examples in which integrins are really helpful. One in migration, as we said before. So white blood cells, these immune cells in our body, travel to infection sites through the bloodstream. That's kind of the highway of the body. And so they are floating around through our circulatory system, and at some point they need to exit the blood vessel at the site of infection. And so what happens is these integrins are expressed on the surface of the white blood cells, and they help these white blood cells to attach to the inner wall of the blood vessel where that infection site is present. And that helps them then to squeeze through the blood vessels, kind of in between the cells of the blood vessels themselves, and to escape out of the vasculature. It is really cool. I recommend you to look up any video about this. These animations, they show the integrins, the white blood cells kind of rolling along inside the blood vessels, expressing these integrins, getting caught on a site, and then squeezing through cells of blood vessels. So in the disease leukocyte adhesion deficiency, basically, these cells cannot attach to the blood vessel and get out of the circulatory system. And so these people have trouble fighting bacterial infections because those immune cells can't get to the site of infection as easily. Another example in which integrins are useful is during wound repair for the formation of blood platelets, for the formation of blood clots. And fibronectin is also important for this. If any of you have heard of Cells at Work, another really fun cell biology video. This is an anime that is all about, mostly focused on the immune system, but there's different characters. There's a girl who's a blood cell. There's this guy who's a leukocyte. They're friends, maybe more. There's these really cute blood platelets that come out and make a blood clot when there's an injury, and evil bacterial monsters. So, Cells at Work. Check it out. Now, in addition to collagen, elastin, integrin, and fibronectin, we also have polysaccharides as an important component of the ECM. And these polysaccharides help to form gel-like structures or more cushiony tissues. Here we have a structure of a glycosaminoglycan. So as you can see in the word, glycoso, glucose, sugar, amino, protein, right? So this is a sugar that's going to be in association with a protein. We have a polysaccharide chain shown here, and one thing that you can see that's unique is that there is this added functional group here, right? And so that's going to slightly change the properties of this sugar. We also have a negative charge to this polysaccharide chain. In addition to having this amino sugar, often these are sulfated, and that is going to add an additional negative charge. See, okay, what's so important about having a negative charge? Well, we remember that having a negative charge makes a molecule polar. And if something is polar, it's going to be hydrophilic. So this is a gel-like tissue, right? A gelatinous solution. And so it's going to recruit water because of these negative charges to help with kind of this compressive type ability. Now these sugar chains, these glycosaminoglycans, are further organized into a larger structure called a proteoglycan. And so proteoglycans, proteo, protein, glycan, sugar. These are much larger structures. So the previous amino sugar chain that we saw in this drawing is represented as a red line. All right, so you've got these amino sugars, these polysaccharides that are attached to a core protein shown in green. And then that kind of bristle type structure is attached to another long core protein. And so this is making kind of like a bristly, bushy type shape. And this is going to take up a lot of space in addition to, as we said before, kind of attracting water. So because of the proteoglycans, presence of a lot of proteoglycans is usually going to be associated with a more gelatinous or compressive tissue, right? Tissue that's more squishy. So you should be able to think about different tissue types and say, all right, based on the tissue structure, do I think these tissues are going to have a lot of collagen or a lot of proteoglycans or glycosaminoglycans, right? For example, bone is going to be dense, it's going to be tough. This is going to have a lot of collagen and not as much of those polysaccharides. The far opposite example, the vitreous of the eye, that is a connective tissue. It's very gelatinous, almost liquidy. There's not going to be a lot of collagen in that tissue, but a lot of those polysaccharides and the structures of proteoglycans. In addition to being polar, these negatively charged glycosaminoglycans are going to promote osmosis, right? So sodium is going to be attracted, a positively charged ion is going to be attracted to these glycosaminoglycans. That's going to have more solutes in that tissue, and water is always going to want to flow towards high concentrations of solutes. In addition to allowing kind of a gel-like or compressible, I don't know if that's a word, a structure that is able to be squished, is that they can act kind of as a web or a net or a filter. Right? They've got all those little brush needles, extensions sticking out every which way. And so when molecules are flowing through the extracellular matrix, they can get caught onto those proteoglycans. Right? We talked about paracrine signaling a few chapters ago, and we said that these molecules are secreted by cells, really mostly just are being sent to cells nearby, and they're not long lasting necessarily. And we talked about different ways that these paracrine molecules can be regulated. So we said that the molecules may not last very long outside the cell, or that they may not be able to spread very far because they get caught in the extracellular matrix. And so the component of the ECM that is acting like this net or this web is the proteoglycan component. So if you're thinking about this as being kind of a limiting factor for cell signaling, you can imagine that the extracellular matrix is going to play an important role in cell migration. There is cell migration that is induced by secreted signals. So a cell may have a signal bind to it on one side, and that tells that cell, all right, I need to migrate in this direction towards the source of that signal. And so if the signal is getting stuck in the extracellular matrix and not reaching that cell, the cell's not going to migrate. However, if the extracellular matrix gets rearranged or modified, maybe more of those signals could come from that direction. It's a really cool idea, and it's something that I'm really interested in studying in terms of helping cells migrate that are transplanted for therapeutic use. So the research that I do with my zebrafish is I'm looking at how does the extracellular matrix promote or inhibit cell migration towards a site of injury. So if you wanted to transplant stem cells and get them to go to a certain place, how can we make the environment conducive to that? All right, so let's do a problem. What do you expect to be the predominant component of the ECM in the following tissues? Tendon, discs of the spine, skin, connective tissue surrounding lungs. Right? So we remember that the more tough tissue needs to be, the more collagen that you will find, the more gelatinous or the ability to compress is going to be due to proteoglycans, and the ability to be stretchy is going to be promoted by elastin. So tendons, those tend to be, they have some elasticity, but not a lot. They are going to have a lot of collagen, but probably not as much as bone. The discs of the spine, if any of you have back issues, you know that those need to compress a little bit. They're acting as cushions in the spine. And so those are going to have more proteoglycans in them. Skin, I would say probably a fair amount of collagen, right? Because we want it to have that tensile strength, but not as much as, say, tendon. It wouldn't surprise me if there's also some elastin in there as well. Lastly, connective tissue surrounding lungs. Right? So there is connective tissue around the lungs, but we know that the lungs move a lot, right? In and out. They need to be able to expand and compress and expand and compress. And so in this case, elastin is going to be really critical for that connective tissue to allow it to move with the lung tissue. Let's talk about objective 2, an example of a tissue and how it is organized. We're going to look at epithelial cells, and then we're going to talk about cell-cell junctions that are important for tissue formation. As we said before, epithelial cells tend to have a lot less extracellular matrix. The cells often tend to be bound directly to each other, and they can be seen in tissue structures such as sheets acting as barriers. And again, this is due to those cell-cell junctions. So anytime you think of a body lining a cavity or covering the surface of something, you are looking probably at epithelial sheets. They, for example, on the surface of the body, these are important for acting as a protective barrier. On the interior of the stomach, also an important barrier. However, sometimes they do need to have a transport function. We looked at earlier, we talked about gut epithelial cells. These intestinal cells are acting as a barrier, but they are selective. And we talked about how they need to let glucose in a very organized manner. These sheets need to be able to stretch. Your bladder, for example, is basically a bag made out of epithelial cells, and it needs to be able to stretch when the bladder is full and get smaller when it is empty. Some of these epithelial cells will also be secreting molecules. The epithelial cells that line the stomach are going to be secreting enzymes and hydrogen, right, to promote an acidic environment in the stomach. In addition to secretion, they also need to be able to detect signals, right? And so here we have some common features of epithelial tissues. First of all, they usually have a top and a bottom tissue. And when something has two sides, we call it polarized. Right? The bottom of the tissue has a basal lamina, so that's usually called the basal side. And then the other side is often facing a lumen or an empty space, and that's called the apical side. There's a lot of cell-cell junctions. As you can see, these cells are all connected to each other, and there's very little extracellular matrix. I don't expect you to memorize the different types of epithelial tissue structures like columnar or stratified, just to recognize that there is a variety. You can have a single layer, multiple layers. Right? Here we have an example of epithelial tissue in which we can see the apical and basal side. This is a tube in the kidney. And here is the kind of the interior of the tube, all right? So water and other solutes or molecules are flowing through this tube. And so this is considered the lumen, right? So if this is the lumen, that means that this side of these cells is the apical side. And if you look closely, you can see each of these purple circles is a nucleus. And you can see that these are kind of some elongated cells. These epithelial cells are very close to each other. They're arranged in this circle tube shape. And so the opposite side of these cells is going to be the basal side. And where you can see kind of a pinkish line is where the basal lamina would be found. The basal lamina is composed of extracellular matrix molecules. In this example, we are looking at the small intestine, and we have on this side the lumen, right? So this is where food and water and molecules are going to be flowing through the small intestine. And we have villi. The surface of the small intestine is kind of very wrinkled and has these kind of villi poking out. That's promoting increased surface area in the gut. And that's really important because the function of this tissue is to absorb nutrients, such as glucose. So these darker shaded cells that are right on the surface are the epithelial cells. If you look closely, you can see the nuclei are shown in dark purple. These have that simple columnar epithelium structure, right? And you can't see the basal lamina as well in here, but the basal lamina would be lining right underneath this outer layer of cells, right? The last example of a polarized epithelial tissue I'll show is the skin, right? And here we have many layers of cells. These other examples, we had just a single layer of cells. In epithelial tissue, you can see that there's multiple layers of epithelial cells. And the side that faces the air is going to be the apical side, and the side that faces the dermis is going to be the basal side. And you can see the basal lamina right here. In this picture, we have a closer example of an epithelial tissue. We can see that on the apical side, there is a lumen. Here there are some cilia kind of coming out of these cells. And here we have the nuclei of the individual cells. You can see they're pretty elongated. And on the basal side, we have It's hard to see it, but this is where the basal lamina would be found. The basal lamina, as I said before, is made out of extracellular matrix, specifically collagen and laminin. And so in this case, laminin is what is helping those cells interact with collagen and using that integrin protein. So laminin, lamina. Now here we have another example, a drawing of some epithelial tissue. These polarized cells we have on the top facing the gut lumen, the apical side. On the bottom, we have the basal side that is interacting with the basal lamina. And it's important for these cells to be polarized because depending on what side of the tissue you're looking at, you have different functions, right? So when we talked about intestinal cells, the side of the cells that is facing the gut lumen have to have transporters to pick up glucose, right? And the opposite side has to have transporters to secrete glucose in a regulated manner to the bloodstream. In addition, in the gut, we also have cells that are secretory. So in gold or orange, you can see these goblet cells. Their function is not to absorb glucose, but to secrete molecules, mucus, into the lumen of the gut. And so it's very important, right, that these are polarized and that they are oriented correctly so that the mucus is getting secreted into the gut and not, say, into the bloodstream. Now this hopefully is a relatively easy question for you that I have here. So what is it that keeps the membrane-bound transport proteins on the apical side of an epithelial cell? Okay, so why is it that the transporters that take up glucose are on the apical side? We know that there is a fluid mosaic system in the cell membrane. So in theory, these transporters should be able to float all around the surface of the cell from the apical to the basal side. But in this tissue, they don't, right? Why is that? They are unable to do this because of tight junctions, right? The tight junctions are these cell-cell adhesions, and they act like a wall that prevent these transporters from floating freely all around the different sides of the cell. Let's talk more about cell junctions. We've talked before about tight junctions. The other two that we're going to focus on are adherens junctions, or sorry, the other three, desmosomes and gap junctions. Tight junctions, as you can imagine, tightly seal cells together and they create a barrier. In addition to keeping transporters on the correct side of a polarized cell, they also keep molecules from passing through that layer of cells. We talked about how for glucose to be transported, it's dependent upon transport of sodium as well and hydrogen for correct flow of glucose in and out of the epithelial cells. And if we didn't have these tight junctions, not only would these transporters be all over the cell on both sides, but the glucose wouldn't even need to go into these epithelial cells. It would just flow right past them on either side. And here we have an example of that function of tight junctions. In this cartoon, we have a sheet of epithelial cells that are held together by tight junctions. And this is what ultimately is the last kind of stand, the last door that really prevents things from passing through this tissue. They are the HODOR of the epithelial tissue. All right, so here you can see that they have put a molecule, a dye that is dark, into the lumen. And if the dye is able to pass in between the cells, you would find dye on both sides of these cells. And what we can see here is the apical side of these epithelial cells. A tight junction is right here. It's hard to see, but they've pointed it out for us. Here we can see these villi or these little cilia that are poking up on the surface of the cell. And this tracer molecule, this dye, these colored molecules are getting stuck here. They are not traveling on the other side of the cells. See how this part is white? And when they do the reverse, when they put the dye on the other side of the cells, you can see the dye travels as far as it can in between the cells until it reaches a tight junction, and it can go no further. So these tight junctions are going to be strands of proteins that span the entire length of that side of the cell that is interacting with another. I don't expect you to memorize the names of these proteins, but you should know again that these are creating a tight barrier. They are preventing molecules from traveling through this layer of cells, but they are important for organizing proteins in the cell membrane, and that this is not a very stretchy interaction, right? So if you pull hard enough on this, it will rip, right? We'll look at some other cell-cell connections that have a little bit more flexibility. Adherence junctions are going to provide mechanical strength to the cell. And we've talked about this idea before when we were learning about our basic cell structure, how there are cell-cell junctions that not only just help cells to connect, but are also attached to the cytoskeleton inside of the cell. And that allows that group of cells to kind of bear the force of pressure and provide some flexibility to the tissue. The proteins that are mediating the interaction between two cells are called cadherins. And in this example, you can see two cells in gray. They're interacting by an adherence junction. You know it's an adherence junction because the proteins that are sticking out that are interacting are cadherin molecules. And on the other side, inside of the cell, the cadherin molecules are interacting with the cytoskeleton. Most of the time, the cytoskeleton protein is going to be actin. Just like integrins bind to the cytoskeleton to really help anchor the cell to the collagen, the addition of these cadherins binding to the cytoskeleton is going to help really promote the strength of that interaction. And so that's why I say that a tight junction tends to be a little more stiff. If you pull hard enough, it'll just snap. Whereas with cells that are connected by an adherence junction, may have a little bit of give, right? They've got more strength because they have this attachment to the cytoskeleton. The adherence junctions are also important for promoting cell shape. In this picture, you can see that we have cytoskeleton actin that is promoting the structure of these microvilli. And then there's also a band that is running along the apical side of these cells, right? And these are connected to the adherence junctions at the interface between these cells. And adherence interacting with actin is very important during development. And I just want to show you an example. So during development, we have tubes that need to be formed, right? We have different types of structures that need to be formed. And here we have an example of tube formation. And you can see that we first have a sheet of cells, and in pink you can see that actin that's along the apical side of the cell. And when this sheet starts to form a tube, you can see it kind of bends in or invaginates, right? So the sheet kind of starts to bend in, and eventually that top half closes off and it pinches off from the sheet, and we have this tube or vesicle. Now, the actin is acting kind of like a belt. It's tightening here, and that's helping this side contract and keeping this outer side more loose. And so a good way to think about this is if you've ever seen curtain rod, right? Or a ruffled curtain, especially, or a pleated skirt, right? You know that along the top you can kind of um, tighten that, and so that you can induce kind of a tighter top half of that tissue of that sheet and kind of more loose as it goes down, right? And so that's really what's happening here is it's just kind of tightening that belt and promoting this formation of a tube. So I don't expect you to memorize how tubes form, but I just want you to know that adherence junctions are important for organizing tissue shape during development. The next type of cell-cell junction we have are desmosomes. These are quite similar to adherence junctions. They have a different type of cadherin protein that's mediating the interaction, and they're also interacting with cytoskeleton. However, in this case, they are interacting with keratin, or an intermediate filament, right? So adherence junctions are interacting with actin. Desmosomes are interacting with intermediate filaments, particularly keratin and spring. And you can notice that there is this plaque structure that has a lot of linker proteins that help to allow the cadherin to interact with the keratin. We have an electron micrograph image here where you can see two cells interacting bound to each other by a desmosome. So in the space in between these two cells is going to be a lot of cadherin protein. This dense dark area is going to be a plaque, and then coming out on both sides is the intermediate filament keratin. Desmosomes are really important for organizing cells in skin tissue. So I tried to find the least gross picture of a blister for you guys that I could, so you're welcome. Blisters, what do these have to do with desmosomes? So cell-cell connections are what help keep tissues and layers of tissues bound to each other properly, right? Now we talked about cells being connected to each other kind of laterally, but these cells also need to be connected underneath to that basal lamina, right? The basal lamina is like the foundation that really holds that tissue in place. And so if you have disruption of that interaction with the basal lamina, the cells on either side are still connected in a sheet, but that sheet is not attached. It will kind of lift off, and that is when you see a blister form. And the particular adhesions, or the particular type of cell junction that is important for cell epithelial cell attachment to the basal lamina is called a hemidesmosome. So desmosome, we've heard about that before. Hemi is half, right? So in this case, a cell is using keratin and this plaque to interact not with another cell, but with the basal lamina. We said that basal lamina is made out of collagen and laminin, right? And here we can see that rather than using cadherence proteins, it is using integrin proteins to span the cell membrane. And we know that integrin proteins are great for helping cells to interact with the extracellular matrix, particularly collagen. Right? So integrins are going to be commonly found as the adhesion protein in a hemidesmosome, and these hemidesmosomes allow epithelial cells to attach to the basal lamina. The last type of cell junction to talk about are gap junctions. And as the name suggests, it is a gap, a hole that is in between that spans two cells. And this is great for quickly moving things from one cell to the next. These gap junctions are formed by these pores are made out of proteins called connexons. So these are going to allow small hydrophilic molecules to travel through in between two cells And so when you need really fast communication to occur, these gap functions are going to help with that. So electrical signaling that needs to travel quickly through cells can be facilitated by gap functions, as well as metabolic coupling. And so what I mean by metabolic coupling is, say you have a metabolic intermediate that is needed in one cell. If it is connected to another cell by a gap function, there's potential for that molecule, that metabolite, to travel from one cell to the other and help that cell. So an example of the electrical signaling is in cardiac muscle cells. So these muscle cells, they're not neurons, right? But they still use electrical signals. And so this electrical signal is able to spread quickly through the heart muscle cells because they are connected by these gap functions. And that makes sure that these cells are contracting simultaneously. My favorite section, stem cells. Right, so a little bit about my research. I study stem cells, neural stem cells, for the purpose of regeneration of the central nervous system. What I'm looking at particularly right now is promoting stem cell transplant for vision, transplanting stem cells into the retina. And we know currently, this is in terms of stem cell transplants, we have clinical trials going on. However, very, very small percentage of the stem cells that are transplanted into animals or people actually get to where they need to go, survive, and become the cell type they need to be. So I'm really interested in studying how those stem cells migrate, how we could facilitate them surviving and getting where they need to go. And the reason that I use zebrafish is not so much that I love fish, they are cute, I guess, um, but that they are amazing in that they can regenerate their eyes and other nervous tissues. So I used to think, oh, zebrafish, okay, fine, leave them, take them or leave them. And then I learned about how regenerative they are. So not only can you clip off the tail fin of a zebrafish and it will regrow, it could get a brain injury, and within a couple of months, you can't even tell. It doesn't even have scar tissue, right? And same with the eye. And so I'm looking at stem cells in their eyes. They have stem cells in the retina, and we don't. And so I look at how those stem cells in their normal environment, how do they know where to go in zebrafish eye? And by understanding that, maybe we can apply some of that knowledge to our transplant therapies. And who knows, maybe down the line one day we can even repair our own eyes and get somehow induce regeneration in our own eyes. But anyways, so here we're going to talk about stem cells, what they are, and the different sources of stem cells. So tissues are not static. And the majority of the tissues are going to be renewed. The cells within them are going to die and be replaced over the course of your life. Some of them turn over more quickly than others. So when these cells divide, when they die, we have these attachments that need to be reorganized, these structures that need to be maintained, right? And we also have gene signals that are important for telling these new cells what to become, right? If you're here, you need to become this. If you're a new cell over there, you need to become that. There are signals that are sent by surrounding existing cells to these newborn cells that help tell them what to become and where to go. And then they also can remember, there is cell memory as well, right? We've talked about that in the past. How does a cell remember what its parent was? How does it become the same cell type? Now, tissues, we tend to think about cells in isolation, right? Like skin, oh, epithelial cells. But the skin is more than just epithelial cells, right? More than skin cells. We have nerves, we have blood vessels, right? We've got pores, secrete, sweat glands, right? And so just like our skin, a majority of our tissues are actually mixtures of many cell types, right? So there's things that must be in a tissue. So it has to have some type of mechanical strength. Depending on the function of that tissue, it may need to be really tough, it may need to be stretchy, right? It may need to be squishy. All cells require nutrients and oxygen and waste disposal. And so in mammals, that is taken care of by the circulatory system. Majority, a lot of tissues, more than you would expect, are innervated by nerve cells. For example, there are nerves in your stomach and your gut that help tell you when you are full. We have immune cells that are important for preventing or acting upon infection that occurs in the cell, and also just taking care of cells that may have died of natural causes as well. Now, during development, I don't know if any of you are taking embryology with Dr. Myers, but during development, we also have a lot of cell migration that's occurring, right? And so you have cells originating in one place and needing to go to another during development. Now, we often think of stem cells just as during development, right? Embryonic stem cells, but there are also adult stem cells. Any tissue that needs to regenerate or has some type of renewal or that is growing is going to have adult stem cells. However, these stem cells are much more restricted in what they can become. Embryonic stem cells can become almost all cell types in an organism, whereas, say, a brain stem cell is an adult stem cell that's only going to become neurons, glia, astrocytes, or oligodendrocytes. Right? So they have more restricted fates. Just to emphasize this point that the tissue is really kind of a community of a variety of cell types. In the skin tissue, we can find that there's epithelium, there is fatty tissue, there are nerves, and there are blood vessels. There's also precise organization of how this is all arranged. In the epidermis, we also have immune cells, pigment cells. In the dermis, this is going to have a lot more collagen. You're going to see more vasculature, right? And then when you get to the dense connective tissue of the dermis, you're going to see a lot of fibroblasts and a lot more collagen. So you can see again that this tissue has a variety of cell types and they are organized in certain locations, right? And so cell communication is really important for these cells to maintain this organization. So any new cells that are born are going to receive signals that tell them what to become and if they need to move, where they need to go. It's also going to make sure that if you have cells that aren't needed, that they don't survive. In addition to telling cells what to become, you can also promote cells' organization by what they can attach to, right? So there are different types, say, of cadherin proteins, right? So I'm just going to take a moment here. In adherens junctions, we know that there are cadherins, right? But not any cell, not any epithelial cell can bind with just any epithelial cell. So of those cadherin proteins, there are different subtypes. There's E cadherin, V cadherin, N cadherin, right? And these cells will only form adherens junctions with other cells that have the same cadherin subtype. So a cell that has E cadherin in the adherens junction will only bind with a cell that has also E cadherin in its adherens junction. And so that would promote those cells to bind to each other. And so if you have a new cell, it's going to see who is my partner, who is the same as me, right? And that's where it's going to attach and where it's going to reside. And so selective cell adhesion is going to help organize tissues. As we've also talked about, we have cell memory, right? So there are transcription factors, these activating transcription factors that can get passed down in the cytoplasm during cell division, and that can then promote more of synthesis of more of that same transcription factor. And that master transcription factor can turn on sets of genes that will tell the cell what to become so that it becomes the same type as its parent cell. Right? So cell communication, selective adhesion, and cell memory are important mechanisms for organizing tissue as it renews. As I said before, some tissues turn over quite rapidly and others much less often. Nervous tissue, we talked about how neurons terminally differentiate. They go into G0, right? A lot of nerves don't divide once they're born. And an opposite example, the intestinal epithelium is replaced every 3 to 6 days. Other examples would be skin or hair. And so this is a little bit of a better understanding behind radiation. This allows you to better understand how radiation sickness works. I don't know if any of you watched the show Chernobyl on HBO. It showed some pretty dramatic examples of this. So your epithelium needs to be replaced every 3 to 6 days. And what radiation is going to do is it's going to kill all those rapidly dividing cells. And so not right away, but 3 to 6 days later, when those new cells are not there that were expected to have been matured at that point, then you start to experience diarrhea and water loss. Right? So it takes a few days to see the effects of radiation. Because cell proliferation and cell loss is so important, right, and kind of irreversible, we have elaborate regulatory mechanisms in our bodies to control this. When these get out of balance, we talked about an example such as radiation sickness, you can also get things like cancer, right? Too much cell production, not enough cell loss. So let's look a little bit in more detail at how stem cells actually produce new cells. In this example, a stem cell divides, and when a stem cell divides, it can either make more of itself, more stem cells, or the offspring can mature and produce a differentiated cell. So differentiated means a mature cell that can perform all of its functions. A stem cell is rather undifferentiated. It has a lot of potential as far as what it can be and do. And so in this example, at a certain point, we had asymmetric cell division occur right here. Right? In asymmetric cell division, one cell type is going to become the same as its parent, and the other is going to differentiate. It's going to become something else. Now, interestingly, you can see that this cell doesn't necessarily automatically mature into its differentiated form. This is on its way to maturing. It's becoming more specified in what it can become, but it still has some proliferative ability here. And so you may have heard of progenitors or precursor cells. So you can have progenitor cells that are they have a specified fate. They're only going to become one cell type. They are still proliferative. Okay? So the key thing that I want you to know from here is that a stem cell can undergo asymmetric cell division, and it can produce one cell that's going to become a stem cell, the same as its parent, and another one that's going to differentiate. I also want you to understand what differentiated versus undifferentiated means. All right, this is just kind of summarizing what I've said. I want to look at an example of replacing cells in a pretty elaborate tissue, right? So here we have gut, small intestinal small intestine epithelial tissue. And we've seen pictures of this before. You can see the epithelial cells are lining this villi, right? You can't see it well. The basal membrane would be right underneath this outer layer of purple shaded cells. And these cells in the epithelium are going to be absorptive, so some cells that are going to be taking up molecules such as glucose. And then you're also going to have some cells, as we said before, that are going to be secreting molecules like mucus. And so these secretory cells are shown in more of a pink color, right? And in this example, the cells need to be replaced every 3 to 6 days. And the cells, as they age, they travel up this villi. And the oldest ones are going to shed off the top here. And so down here, these are called crypts. This is where the stem cells, the adult stem cells, reside. So these stem cells are going to divide. These proliferating precursor or progenitor cells are going to travel up the side of these villi, and they're going to mature as they travel upwards. On the right, we have a diagram that demonstrates this in more detail. In another example of cell replacement, we talked about how epithelial cells, these have multiple layers of cells, right? In this last example, it was just one sheet of one layer of cells. Here we have multiple layers, and this is kind of a not perfectly flat tissue. You can see it's got some wrinkles kind of in it. And you can see we've got epidermis and dermis in here. And the epidermis contains stem cells in the basal layer. So in the bottom layer of the epidermis, along here, are going to be the stem cells, the adult stem cells. Right? And then as they mature, as they differentiate, they're going to rise in higher and higher layers. And eventually they're going to die, and those dead cells are what are on the surface of our skin, and they eventually get shed off. You can see in more detail here a diagram showing the basal lamina, where the stem cells are formed, and in what direction they're traveling as they mature. We talked about how cell signaling is important to maintain organization of a tissue. Right? So first of all, you know that cells will receive signals from their surroundings that will tell them when they need to divide. Right? So say if there's an injury or if a bunch of cells get old and die, there are going to be signals that tell those stem cells that they need to divide. And one example of such a signal is Wnt, the Wnt signaling. Wnt is one type of extracellular signaling molecule, and I don't expect you to memorize how Wnt works in the intestine. This is just an example for you to really see at work how a signal can induce stem cells to divide and also to promote organization or to maintain the organization of a tissue as these new cells are introduced. We've got our villi shown here in the cartoon on either side. This layer of epithelial cells containing absorptive and secretory cells. And in the crypt, we have the stem cells shown in red. Again, these stem cells are going to respond to an extracellular signal, Wnt, and that's going to tell them to divide. And then as they mature, they're going to migrate up this villus. And it's pretty neat because Wnt is found by cells only, it's secreted by cells only in the crypt. So only cells that are down in that area are going to be proliferative, and cells that are up further high on the epithelial tissue on the higher up the villus are not going to be proliferative because they are not near that signal. So these signals are also, in addition to telling the cells to proliferate, they're going to tell these new cells whether they're going to be secretory cells or absorptive cells. So you can see, again, secretory cells in the crypt are going to produce Wnt. Wnt is only found down here. It's kind of loses concentration as you get further away. And so only cells in the crypt are going to be proliferative. And you can have a disruption of this signaling process. And when that happens, you can have overproliferation that can lead to cancer. In some tissues and in some organisms, stem cells can repair damaged tissue. And in humans, a good example we have is the use of stem cells for diseases of the blood, such as anemia, really bad infections, and leukemia. So in these systems, in these diseases, you've got blood cells that are not functioning properly. And so the way that this works, this is really the first real successful use of stem cell therapy, and it's bone marrow transplant. What the researchers or doctors will do is that the first thing that they'll do is remove all of the dysfunctional or damaged blood cells, and then they will replace those cells with a donation of healthy blood cells. And in this donation is going to be stem cells, blood stem cells. And so eventually those blood stem cells will divide enough to repopulate all of the lost cells. So typically irradiation is used to destroy the damaged blood cells. A bone marrow transplant is performed, and in the bone marrow is where the majority of the stem cells reside for the circulatory system. These new blood cells, as I said, are going to have stem cells in them, a significant proportion of that. And so those are going to divide over time, and they are going to basically give that person a whole new set of blood cells. And the idea is that now they've removed all the cancerous or the infected cells, and that this person could move on and have a healthy body. In any type of stem cell transplant, whether you're doing bone marrow or whether it is a embryonic stem cell transplant, you have some common obstacles that must be overcome. The first one that you've all probably heard of is immune rejection. So you need to make sure that, for example, if you're donating bone marrow, that you have the same blood type, right? And even if you are the same blood type, there is still a possibility that your body will say, this is not my tissue, and the immune system will attack those donated cells and kill them. And so people that get transplants often are on high immunosuppressives. Another obstacle is the source of the stem cells. So sometimes it can be hard to get those stem cells. In other cases, it may be ethically controversial. So bone marrow are adult stem cells. There's not too many people that argue that there's an ethical issue with that. However, embryonic stem cells, there is a lot of controversy surrounding the use of those, because in order to get those pluripotent stem cells, you have to destroy an embryo. And if you view that embryo as a person, as a life that deserves protecting, it's equivalent to murder. Now, on the other hand, you may say, think of all the people that are dying because they're not getting those embryos, right? So just a lot of controversy there. Another example of a source of stem cells, we have adult stem cells, right? And so people have been looking at using umbilical cord stem cells for transplants, and there's been a lot of really great research coming out of that. And again, for example, we do have in our brains small regions of regenerating cells. The majority of the brain does not divide, but there are some proliferative regions. Now, it would be really hard to donate neural stem cells because you would have to drill into my brain and get into my hippocampus, right, or into one of my lateral ventricles to access those stem cells. It's just not feasible, right? So immune rejection, the source of the stem cells, and then lastly, are those cells going to become what you want them to become? So if you inject, say, embryonic stem cells, there is the possibility that they could get signals from their surroundings and become the cell type that is desired. But they could also, because they are pluripotent, they can become anything. So only if you have a strong stomach should you Google the tumor, the cancer teratoma. This is a cancer in which the cells become all sorts of different mature cell types. So you can find teeth and hair in these tumors. They're just really gross, all right? And so often what scientists do now, rather than implanting embryonic stem cells, is they will induce these cells to partially differentiate. So remember when we talked about stem cells and how we can have stem cells and precursor cells before you get to the point of mature cells. Scientists are learning what those signals are. So rather than say, we're going to implant embryonic stem cells into your brain, they would partially differentiate those cells into neural precursor cells and then implant those into the brain. And now they're much less likely to form these tumors. We're still trying to understand what those signals are that tell a cell what to become. And so that's a really active area of research. Whether you're using adult stem cells or whether you're using embryonic stem cells, this is a really important factor in to consider for successful transplant therapies. So my cats have just come into the room and they're making some noise, so sorry if I'm a little distracted. Let's talk a little bit more about the different sources of stem cells. So as we said before, embryonic stem cells are pluripotent. They have an unrestricted potential in what they can become, and they have been used in research with a lot of success for diseases, a variety of neurodegenerative diseases, as well as heart diseases, spinal cord injury. The ones that I've underlined are ones that when I looked a year or so ago, that there are active clinical trials going on. Eventually, we may be able to create whole organs from stem cells. So in this example, a research study was done in which they used embryonic stem cells to form like immature retina with all the different cell types that are found in the retina organ and all the different layers, which is pretty cool. As I said before, embryonic stem cells can become all cell types, and I just wanted to briefly go over where they come from. So when egg and sperm unite, they create an embryo, and this embryo during early stages is a kind of an outer sphere of cells, and then inside of that sphere we have this kind of mass of cells, the inner mast cells. These are the cells that are the pluripotent cells. If you ever have testing done, say during IVF, they will take some of these surrounding cells to do genetic tests on them so that they don't damage this inner mast cell. So these inner mast cells have to be removed, and that is what destroys the embryo. And then these inner mast cells can be used for potential regeneration of all sorts of tissues. All right. Now I'd like to talk about induced pluripotent stem cells because a lot of people are unfamiliar with this, all right? So check this out. You can take mature differentiated cells, such as skin cells, and in a dish induce them to be just like embryonic stem cells. So you can de-differentiate these mature cells to become pluripotent. This is pretty amazing work, all right? The guys that discovered this won a Nobel Prize. If you remember when we talked about cell memory and gene expression, we talked about master transcription factors. And we showed an example where these scientists, they figured out what the transcription factor was that oversaw eye development, right? If you turn on this one transcription factor, it activates another set of transcription factors, which then activate another set until all the genes that are required to form an eye have been turned on. And so just like that, scientists were able to identify 3 key transcription factors for inducing any type of mature differentiated cell into this pluripotent state. And so it's not of embryonic origin, so it's not called an embryonic stem cell, but it is pluripotent, and it's a cell that's been induced to become pluripotent, hence its name. Those 3 transcription factors, if you're curious, are OCT3/4, SOX2, and KLF4. Right. Now, the thing that is really nice about this is that you could treat yourself with your own cells, right? So rather than having this potential immune rejection due to transplant of stem cells from another individual, these originated from yourself, and so they have your own self markers on them. So your body sees those cells as its own, and the immune system is less likely to be activated. So there's research where people have said, or what the goal is that you could, if you needed a stem cell transplant, that you could isolate some of your own, say, fibroblasts in a dish. Those could be differentiated into this induced pluripotent stem cell fate. Then they could be further matured using other factors to become, say, a retinal progenitor or a neural progenitor or a cardiac progenitor cell, and then transplanted back into you, and you would not reject those cells. Now, there is still a lot of work that needs to be done in this area. So first of all, it's a lot of work to get those cells into that pluripotent state. So the rate is very slow. And also what you've done now is you are proposing that you are inserting cells that have been genetically modified. And so there's always concern that there were some off-target effects, right? And so gene therapy, as you know, is kind of it's been coming for a long time. And it just is going to require a lot of extra work to show that these changes are specific. So, in addition to replacing lost cells that were lost due to damage or disease, there are other important uses of embryonic stem cells and induced pluripotent stem cells. So if you wanted to get a bunch of cells of the same type to study, say, how a disease works, the embryonic or induced pluripotent stem cells are a great source, right? I'd like to show you an example in the case of Timothy syndrome. This is a genetic disease that is caused by a mutation in a calcium channel. And there are a variety of symptoms in different organs, such as abnormal heart rhythms and autism. Now, if I wanted to study this, I'm not going to be taking neurons from a person, right? That's going to cause problems, right? Same with isolating heart tissue. You know, that's pretty important tissue. Now, instead, what you could do is you could isolate fibroblasts from a person that has this mutation, induce those fibroblasts into stem cells, and then tell these stem cells to become neurons. And now you can study the molecular mechanisms behind the autism, for example, due to this genetic disease, in basically this person's own neurons without having to actually take neurons from their brain. Pretty neat, huh? So again, you would derive iPS cells from a patient with a disease, and then you would differentiate them into whatever tissue type that you want to study. And then not only can you test out how the disease works, but if you wanted to test out candidates of medications, you could do that as well. And you've just got a ton of cells, right? As I said before, it's important that you're able to compare and contrast adult-induced pluripotent adult stem cells, induced pluripotent stem cells, and embryonic stem cells in terms of what they can become, where they come from, the different types of ethical issues that are associated with them, right? Things like that. For our last objective, we're going to talk about cancer. Again, this is a disorder of renewal and organization of the tissue. So cancer cells, some of their main properties is that they're very proliferative, they're invading tissue, and they're traveling to places where they shouldn't be. And this is due to a genetic mutation that's going to allow a cell to survive in places that it shouldn't and divide when it hasn't been told to divide. So it's kind of like a rebel. It's just going off and doing its own thing, and it's not listening to its community, right? And it divides, and the cells that it produces are the same thing. So now it's got a whole gang of buddies that are not listening to what they're being told. Now, you're thinking, okay, too many cells. What's so bad about that? Well, think about the structure of some of these tissues, right? They're very organized. And so if you have a clump of cells of the wrong cell type in the wrong spot, that can prevent that tissue from functioning properly. So there are some properties that are inherited by these cells when they proliferate. First is proliferating when they shouldn't. And then second, invading, going where they're not supposed to be. So if you've heard someone say they had a tumor but it was benign, that means that the cells are dividing more than they should, but they are not traveling throughout the body. And then the worst case is when they are malignant, and when people say the word malignant, what they mean is that these cells are now traveling to other tissues throughout the body. On the right, we have a cartoon demonstrating over-proliferative cells that have now started to invade. So see how they have escaped. These are epithelial cells in this example. They have escaped from their tissue. They've traveled past the basal lamina, and now they've gotten into the vasculature and are able to travel through the body. In this example of a liver on the right, this individual did not have cancer that originated in the liver, but those cancer cells eventually made it to the liver and then formed tumors. So these are called metastases. So how do cancer cells develop? Often it is not a single mutation that causes cancer, but a accumulation of mutations. We know that mutations, things that cause mutations are called mutagens, right? And even without mutagens, you can have mutations arise. Anytime a cell divides is an opportunity for a mistake to occur, because every time that a cell divides, it has to go through S phase and replicate its DNA. And even though our DNA polymerases have proofreading mechanisms, they are not perfect. They're not 100% perfect. And so there is an opportunity every time a cell divides for its DNA to acquire a mutation. So you're thinking, how do we all not have cancer, right? Well, first of all, those DNA repair mechanisms that we have, the proofreading is good, pretty good. We have repair mechanisms that will go and check and make sure that the cells, say, will not divide if there's DNA damage. They will also go back and fix. So we've talked about nucleotide excision repair in our LitLab 2, right? But you do notice that as people live longer, there are more rates of cancer, right? So the reason why cancer rates have gone up is because we are all living longer. We just didn't live long enough to acquire enough mutations to cause cancer in the past when people only lived to, say, be 50. Now, I want to talk about how cancer cells can evolve, all right? So this is one reason why cancer can be difficult to treat, all right? So say you get a mutation and it happens to be not just in some random gene, but in the DNA repair system, all right? So not only do you have a mutation, but now the system that is supposed to fix mutations is broken, all right? That means that they are going to start getting more and more mutations even more quickly that aren't fixed, right? And so the more genetic diversity that you have, that a cell or an individual has, the more opportunity there is for survival, right? And again, there's as more mutations are acquired, the cell now has more abilities potentially to resist different treatments or harsh environments. So if you remember learning about natural selection and evolution, it's quite similar. So in a population, in a species, for example, if you have diversity, there is more likely a chance that that species as a whole will survive, because even if some of them don't have the beneficial traits and die out, others, because of that diversity, may have some trait that gives them an advantage. They survive and they pass that trait down to their offspring. Over time, that population now all will have that beneficial trait. The same thing is happening in these cancer cells. But not only that, they're acquiring mutations at a much more rapid pace, right? When we're talking about evolution of species, right, these mutations don't occur very often. The more mutations you have, the more opportunities to survive any threat that you are met with, right? Oh my goodness, cat. Right, so any mutations that are going to promote cell proliferation, survival, say not listening to cell death signals, or enhanced migration are going to be selected for. In this example, we have a cell that acquired a mutation, gave it a slight advantage, and maybe it then acquired a second mutation that increases that advantage. And then with time, that cell may be, maybe that mutation was in a DNA repair mechanism or a proofreading mechanism. And so now it is even more likely to get mutations. And one mutation it happens to acquire is something that makes it invasive, all right? Now this cell can migrate, it can make more cells like it, and can travel throughout the body. We like to say it's a collection of subversive behaviors, right? These cells are like rebels in the tissue. They don't need as many signals from other cells to survive. They also don't listen to them as well. They are going to survive in environments that are harsh. So for example, in the center of a tumor, there's not going to be a lot of blood flow, not much vascularization. And so normally cells would die in that case. Instead, the cancer cells evolve the ability to survive in low oxygen environments, right? Again, they're proliferating indefinitely. Cells that normally would stop dividing are not. They're also genetically unstable. So as I said before, if you get a mutation in something like a DNA repair gene or a checkpoint protein, this cell is more likely to acquire additional mutations. These cells are abnormally invasive. We talked about how cell-cell adhesion helps cells know where to go in a tissue, right? these cells will change what their adhesion markers are so that they can travel to places normally they weren't allowed. They'll also have abnormal metabolism so they can survive on reduced nutrients if needed. And again, they're able to survive in places that they normally wouldn't. The next thing I'd like to talk about in terms of cancer formation are oncogenes and tumor suppressor genes. These are both genes that can cause cancer. So normally these oncogenes and tumor suppressor genes, their jobs are to maintain the cell cycle, right? So they'll tell the cell, yes, it's okay to move on to the next stage of the cell cycle, or they may be signals that tell the cell to stop, it's not time yet to divide. Oncogenes, these are genes that normally tell a cell to proceed through the cell cycle, right? So these are going to be, say, checkpoint proteins that tell the cell, all right, we've got a signal to divide, let's move on into S phase and replicate our DNA, for example, right? So that's good, right? But if this gene gets a mutation and it gets stuck on, it's going to tell the cell that it needs to move on into the S phase, even if, say, the cell never received a signal to divide, all right? So this is like in a car if you've got the gas pedal stuck down to the floor, all right? You can't stop. The other type of gene that we're going to talk about are tumor suppressor genes. So these are genes that usually tell the cell to stop, to not progress through the cell cycle. We talked about genes that sense whether or not there's DNA damage. They sense whether or not the DNA has been divided properly during mitosis. And if something is not right, they are like, they put on the brakes, right? And they say, stop. We don't want to go further until an issue gets fixed. If this gene gets inactivated, it's like your brakes stopped working, all right? And so you cannot stop moving if you're in a car, right? So both are bad. Not being able to stop, or not being able to, if your gas pedal is stuck down, right? Or if you can't get your brakes to work, if they're broken, right? Now, so in oncogenes, typically we're talking about gain of function mutations. So this gene is not shutting off when it's supposed to. And in tumor suppressors, we're usually talking about loss of function. This gene is not able to tell the cell to stop dividing. And because of that, I want to get into there's some genetic implications that I want to get into. If you remember, we have diploid cells, 2 sets of chromosomes. And so when you have a gain of function mutation, as long as one of those alleles gets mutated, you're going to see an effect. Now in the tumor suppressor case, this is a loss of function. You have 2 alleles. If one gets inactivated, you still have another copy that's functional, and so your body is okay. You're not going to get cancer. However, you are at an increased risk for cancer because now you only have one, you have used up your one chance, right? Kind of like 3 strikes, you're out. And so that's what is being shown in this picture. So some people may inherit a genetic predisposition for cancer and that one of their alleles has the mutation, but it's a loss of function mutation. And only if at some point they somehow acquire a second mutation, say from the environment, will they actually get cancer. Again, on the opposite side with these oncogenes, this is a gain of function mutation. You only need one allele to get it, and so you're much more likely to get cancer in this case. Since we are in cell biology, I'm going to dive a little bit deeper into the mechanism of these tumor suppressor genes. In this example, we have a mutation in one allele on the left that was inherited, and another allele is normal, all right? So this person is at an increased risk, but they do not yet have cancer. And there are a variety of ways that they can basically get to this point where they are likely to have cancer. So we've talked about gaining a mutation in the other allele. So we can see that in this third example here, or in this second example, there's a gene deletion. If, for example, the paternal chromosome is lost, you've lost your second chance, that cell is going to become cancerous. Now, we've talked about this idea of epigenetics. This was introduced in cell biology. These are changes in gene expression that are not actually due to mutations, right? The gene is, say, tightly packaged, so it's not accessible, or it has been modified so that it is more accessible to, say, transcription. Now, in this case, it's a loss of function mutation. So if you have an epigenetic modification that occurs in which that gene gets tightly packaged and is not accessible for expression, it's not available for transcription, it's just the same in effect as acquiring a mutation in that gene where it can't function, right? So in addition to mutations, you can also have epigenetic changes that can cause cancer. So pretty complicated. There's some main fundamental pathways, however, that you can focus on when you're thinking about what causes cancer. These are going to be alterations in the pathways that regulate cell proliferation, obviously. DNA damage response, really important, right? Because if you have mutations in that, now the cells are going to acquire additional mutations at a faster rate. And then third, alterations in cell growth. I'd like to give an example. We talked about colorectal cancer, or no, we didn't. We talked about epithelial cancer before. Colorectal cancer, it's also an epithelial cancer. And this is one that is associated with old age. Majority of people that have colorectal cancer did not inherit a mutation for this. Though there are some cases in which people in their family the family is known to be prone to the disease. So there is, again, the majority of colorectal cancer does not have a hereditary component, but they have identified some genes that are important by studying families that have a propensity for cancer, right? So these people, because it's a genetic there's a genetic cause for this that's inherited, they tend to get this cancer earlier in life, and they can identify that these people are likely to get cancer due to polyps or overgrowth of cells in the epithelium. Interestingly, they found that the formation of these polyps is caused by a mutation in the gene it's the end of the semester. I don't want to pronounce this. Adenomatous polyposis coli. All right, we're going to call it APC. All right, either deletion of this gene or inactivation. All right, so this is a loss of function mutation. So what happens? We're thinking here, okay, loss of function, tumor suppressor, perhaps. These people in this family, they are inheriting one mutant copy and one normal copy of the gene. So you could say they're heterozygous in a way for colorectal cancer. So they are at an increased risk for cancer because they've already lost their one chance, right? Versus other people that are not born with this mutation. And so that is why you're going to see people that inherit this are going to get cancer much earlier in life as well, right? So by researching these families, they were able to identify this APC gene that was mutated. And they found that this was actually important information for people that the majority of other patients that didn't inherit this mutation. They were born with two good copies, but at some point later in life, this gene acquired mutations and was inactivated. And so a really good example of how people volunteering to participate in research has made an impact, because now we can better understand how the majority of people, for example, how colorectal cancer works in the majority of people. All right. I'd like to talk a little bit about treatments for cancer. And so hopefully by getting through this lecture, you're getting a better understanding of why there's likely not going to be a one drug for all cancers, right? You can have the same gene be mutated 100 different ways, right? Or you can have unique combinations of genes be mutated. And so again, it's not always one gene is the cause for the cancer. And so being able to better understand on a case-by-case basis how this cancer formed, you're better able to personalize treatment. So some of the challenges that doctors and researchers are working on is being able to classify cancers. So to be able to identify, oh, if you have this set of genes mutated, this is going to be an aggressive cancer, versus if it's this set, it's going to be maybe more slow growing, or these genes are going to be associated with metastasis. And that way they can better customize the treatments that are used. When I was in grad school, this was kind of one type of research that I did. I worked in a lab that studied bladder cancer, and they looked at bladder cancer caused by heavy metal exposure. Bladder holds a lot of waste, so those cells are going to be exposed if you do ingest or are exposed to heavy metals. And so that is one way that bladder cancer can occur, from environmental heavy metals. And so we were looking at different genes that could be used as markers of metastasis. So the people in my lab, they did a microarray, and they looked at all these cancer cells, and they looked to see what genes were commonly overexpressed or underexpressed. And then they looked to see if those genes correlated with metastasis in mouse models and things like that. All right, so I'd like to finish up with just talking about some strategies, some exciting new methods that are coming out to treat cancer. So first, we know that these cancer cells are genetically unstable, right? They are constantly they're more frequently acquiring mutations, but there is a point where cells cannot handle it anymore. And so one approach is to just increase the amount of mutations, the genetic instability of those cells to the breaking point and where the cells will die. A second strategy that you may have heard of in the news is using the immune system to kill tumor cells. So what if we could tell our immune cells that these tumor cells are foreign, that they are not our cells, and that they need to be killed? That'd be pretty cool. And so what scientists do is what they've been doing is identifying the surface markers of cancer cells and then injecting those proteins or those molecules into a patient. And what happens is the hope is that your immune system would be activated. They would identify those as harmful, right? And that the immune system would attack other cells, such as the tumor cells that are also expressing those markers. The last strategy that scientists are using is by better understanding the signaling pathway, is to target the signaling pathway of these cancers. So if they know, for example, an oncogene, a gene that's overactive, and they know how that gene normally works, what mechanism it's using, maybe they could target something downstream of that oncogene to inactivate that pathway. And so one example is Gleevec, that is countering an overreactive signaling pathway. That is it. I'm sorry I went a little over today. I hope you enjoyed this chapter and this class. I will continue to be finishing up reviewing your reports, your cell cell culture lab reports, and I'll make an announcement about it on Canvas because I'm sure not everybody made it to the very end of this lecture. But I have been editing your papers, your first drafts on Canvas in SpeedGrader. And so you should be able to see my comments if you pull up a PDF of your assignment. And in the comments section, I've attached a rubric that will show you kind of where I took off points. I tried to grade pretty easy on this. I basically, if you attempted to do something, you got the points. But if you missed something completely that was listed on the rubric, you may have lost some points. Obviously, I'll grade stricter for the final report. You may have noticed that I extended the deadline to the 23rd. So don't submit it late because that's the very last day of the semester. I can't accept late submissions. And follow that rubric carefully, all right? Best of luck on your final mini exam or final exam. There is no final in this class. Woo-hoo. And with Lit Lab 3, that is the last Lit Lab of the semester as well. Please feel free to email me if you have any questions, and I will see you around. Have a good summer.